There are several things scientists need to do at the computer, but today I'm going to focus on one of them, or two, but which are both related to one thing, and that is programming. I was never super interested in programming, nor did I ever think I would need to learn it when I started studying biology in my bachelor's. But then reality happened. The first time I really had to deal with any sort of programming was during my master thesis. During my master thesis, I was studying marine birds and their brooding behavior, which if it's something you want to learn more about, you can check this super ultra cringy video of Maria from the past. But long story short, I was trying to understand the influence of the lunar cycle on how many times the parents would come to the nests to feed their chicks. Scientifically, whenever you want to determine the influence of something on something else, or when you want to find correlations between different variables, you need to perform statistical tests. These tests vary in type and in complexity depending on what it is you are trying to assess and how many variables you want to include in those tests. But they are necessary. Otherwise, you might think you see a pattern that actually does not exist and is simply a product of your human brain seeing patterns where they do not exist. Or on the other hand, maybe you don't see a pattern that exists, and testing and modeling might help you find it. And there are several ways you can do statistics and statistical tests. I mean, statistics dates back to the 17th century, a time when computers were nowhere to be found. Which means you can technically do it by hand even. But nowadays we use computers. And that's where statistical programs come in. To analyze the data I collected during my master thesis, I used a program called R. And during that time, I learned R language, which is the language you need to operate said program. By applying lines of code, you can do tests, create plots, create models, and do a bunch of other cool data analysis stuff. Back then, I didn't really need to learn hardcore programming. Most of the tests I wanted to do were very basic tests that had already been hard code programmed.
Does not compute.
I think I said that right. And I just had to apply them to my specific data set. But in order to do that, I still needed to understand a bit of the language itself. So I did learn the basics of the R language. Learning R for statistics was, oh, sweet summer child. Fast forward to my PhD and I met the final programming boss, bioinformatics. Bioinformatics is what happens when computer science and biology merge. According to the National Human Genome Research Institute, bioinformatics is a field concerned with the acquisition, storage, analysis, and dissemination of biological data, most often DNA and amino acid sequences. One of the things I was trying to figure out during my PhD was whether there are bacteria in the ocean that can degrade plastic. If you want to learn more about that, also check out this video, which is not so cringy, about my PhD. And one of the things I did to figure that out was looking at what type of substances some of these bacteria could potentially produce. And to do that, I was studying their genes, which are responsible for the production of said substances. So basically, I was studying their DNA. But how do we do this? How do we look at DNA?
DNA, short for deoxyribonucleic acid, is the macromolecule that carries genetic instructions in all living things. All bacterial cells and most human cells have DNA molecules. The molecule is composed by two strands that wind around each other, forming a helix, and each strand has a backbone made of sugar and phosphate groups. The two strands are held together by bonds between two nucleotides. There are 4 types of nucleotides in DNA: adenine, cytosine, guanine, and thymine, which are normally represented via their initials. Variations in the order of these nucleotides along the DNA molecule is what creates the different genes. To figure out which genes I had in my experiments or in the bacteria I was studying, I extracted them, which just means breaking the cells up and removing the DNA from the cells. And then I sent said extracted DNA to sequencing, which was performed by a company. They do it. Sequencing determines the order of the different nucleotides in the samples that we send. The output, which was what the company then sent back to me after having sequenced
sequenced the DNA samples I sent were files that looked like this. Each file has tens of thousands of DNA sequences that then need to be filtered, further processed, and analyzed by the user, in this case, me. And there are a bazillion tests and analyses that you can do with this type of data. But I will give you one example of what you can do with this type of data based on something I actually did for my PhD. One of the first steps in determining whether there are bacteria in the ocean that can potentially degrade plastics was identifying the species of bacteria that were living attached to plastics that I collected from the ocean. And to do that, I looked at a gene called 16S rRNA, which is a ribosomal gene that exists in all bacteria. This is a gene widely used to identify bacterial species, and therefore it has been characterized for several species, and there are good databases that I could compare my samples to. The sequencing companies, after having sequenced the samples I had sent them, sent me files with 16S rRNA gene sequences. Each file represented one sample, and in each file there were tens of thousands of sequences. To find out which species these genes belong to, I had to compare these sequences to the thousands of sequences in the 16S rRNA gene databases. And as I can imagine, this is virtually impossible to do without the help of machines. Because this is such a widely used method in microbiology, there have been already a lot of methods developed to do this. So I didn't really have to hard code any program per se. I just had to learn the basics of certain programming languages in order to make the programs work with my specific data set. This is only one thing you can do with bioinformatics. In this specific case, in this specific example, I was just looking at one gene at the time. Bacteria have thousands of genes. So you can do all sorts of analysis with all sorts of different genes. You can look at a bunch of different things. While I did not have to learn any hardcore programming, I did have to learn to be comfortable with applying certain lines of code in order to make things work. And to be comfortable doing that, you do need to understand the logic behind programming languages. And which languages you will need to learn will really depend on what you want to do. I lost count of how many hours and days and weeks and months I spent learning all this. When I started my PhD, I had never learned programming. I learned a lot of this by myself. And of course it has pros and cons. The con is obvious. It took me a very long time to get some basic things right. I spent hours trying things and failing, trying things and failing, trying things and failing, without really knowing what I was doing really and why things were failing. Of course, I had people helping me within the group that had a bit more knowledge, but not for my specific data set. There were things I needed to kind of learn by myself. Now, don't get me wrong. Programming is and will forever be a love-hate relationship, but it can be a fun love-hate relationship or a painful love-hate relationship. And in the beginning, for me, was pretty painful because the learning curve was really steep, so steep that I felt like I was just falling back down all the time. But you do reach a certain point in which there seems to be a click in your brain and you realize and understand what is finally happening in this weird thing that you are trying to figure out. So if you are planning and working with any type of DNA or protein analysis or RNA analysis, bioinformatics is going to be on your to-do list. Thank you all very much for watching. Thank you to all my patrons over on Patreon for supporting what I do, basically. And that's it. Thank you very much. If you have any further questions, please leave them down below, and I hope to see you in the next one. Bye.